Yutaka Make is proud to present their new rat repellent, with a unique smell that scares rats more than their natural enemies. Now, let me explain the research behind Yutaka Make's new rat repellent. The rat repellent scent was discovered during research on how scents impact emotional and behavioral responses in mammal neurochemistry. The conventional idea was held that the mammalian reaction to smells was determined based on observation and experimental data. For example, saliva flows when you smell apple pie. However, the research proved that there exist neural circuits in the brains of mammals that determine congenial emotional and behavioral responses to scents that are instinctual. There are two mechanisms that detect smells: the congenial route or instinctual route, and the acquired route or learned route. The olfactory cells of the nasal cavity detect scents that then go through to the olfactory bulb in the brain that interprets these scents. Yutaka makes products that focuses on the congenial or instinctual region that determines these emotional and behavioral responses to scents that instinctually tell rats to run away from their enemies. When rats smell certain substances, their instinctual response is fear. The natural enemy of rats are cats and foxes. When rats smell cats or foxes, there is a freezing phenomenon that occurs, along with other characteristic reactions. Cat urine, a scent of one of the rat's natural enemies, has long been used to repel rats all around the world. The effectiveness, however, of such natural substances has varied according to rat species. As well, the reaction of the rats to the scent was not constant. Now, an innovative chemical has been found which is 10 times more effective than these natural substances. Yutaka Make's new rat repellent is the world's first product containing this chemical to repel rats. The graph shown here compares fox secretions with Yutaka Make's new rat repellent. On the left, fox secretions. On the right, Yutaka Make's new rat repellent. The volatility index, or VIX, represented on the vertical axis of both graphs are the percentages of the time rats freeze 20 minutes after they have smelled the substances. The higher the number, the more the rats are scared. The volatility index of fox secretions is 4%. However, the Yutaka Make's new rat repellent is over 40%. That means our rat repellent is 10 times more effective. Please watch these two video clips where our research assesses the efficiency of Yutaka Make's new rat repellent. First, you will see the general action of a rat bustling normally around the cage. In this comparison, you will see the general rat repellent on the left and our Yutaka Make's new rat repellent on the right, with each respective scent in the upper right corner of each cage. On the left, the rat smells the general rat repellent and goes away from it. The rat on the left continues this same action of smelling and running away until the end of the video clip. Now please watch carefully the reaction to the rat when he smells Yutaka Make's new rat repellent. He is frantically trying hard to escape the cage, looking for any and all methods of escape. Still looking. Still looking. He's still looking for a way out. Now, after he smells the new rat repellent the second time, he defecates. The rat then goes into the corner and for a moment looks for a higher way to get out of the cage. The rat then freezes or shrinks. The rat continues to freeze so that it will not be found by its natural enemies because it could not find a way out. This is the freezing phenomenon. The video on the right is not on pause. In this video clip, you will see the research assessing the efficiency of the new rat repellent. The rat lived in this setup for 1 week prior to the beginning of the experiment to measure the amount of food eaten. The rat's living area, with its rat's nest, is in the central chamber. The tubing connecting the feeding chambers are made of wire mesh to create a neutral scent zone. The chamber on both the left and the right have been fitted with food and water for feeding. Both chambers are vented at the top to maintain each chamber's scent. After 1 week, a dummy atomizer containing water was placed in the left feeding container. In the right, Yutaka Make's new rat repellent atomizer device was placed. Now, prior to the placement, the rat had habitually eaten in the right container. What's going to be the rat's reaction? For a moment, the black rat attempted to go into the right chamber, but quickly then went into the left chamber. The rat stands to get the food. Let's do a slow motion replay. Is he gonna eat? Is he gonna eat? He's eating, isn't he? Then he goes back into the living area. He seems wary of something, perhaps. After a while, the rat goes back through the mesh tubing to the right chamber. Oh, here he goes. After a while, but then he retreats quickly. He goes back into the living area and once again goes back into the left feeding chamber. Let's look at it again in slow motion. Look how quickly he reacts and then freezes from the feeding chamber after he smells Yutaka Make's rat repellent. Here are the test results from the previous experiment. 3 different rats were used in this experiment. Of the 3 rats in the experiment, you can see that rats A and C have 100% effectiveness over 7 days. As for rat B, on the 6th and 7th day the percentages went down. Overall, the rat repellent had an effectiveness of 98.2%. That's right, 98.2%. This does not mean that the percentages will decrease if the rat gets used to the smell. As we look at the chart, the yellow line is the genetically programmed congenial fear of the scent used by Yutaka Make's new rat repellent. The blue line is the fear of the scent acquired in a study using electric shock. In the electric shock study, the blue line continues to decline over time. On the other hand, the yellow line representing Yutaka Make's new rat repellent does not fall but continues to increase over time. The congenial fear reaction of Yutaka Make's new rat repellent does not slow down over time, because it is essential to the rat's instinct of survival.